Hello medical geeks, in today's video we're going to talk about fat acid synthesis. I'm going to explain this topic in 5 minutes. So please make sure that you follow me on TikTok, make sure that you go on YouTube and subscribe to my YouTube channel as well. So let us get started. First of all, we need to start with the definition of fat acid synthesis. So what is fat acid synthesis? Fat acid synthesis can be simply defined as the process by which we make fat acids. And you need to understand that this process is taking place in the cytoplasm of the cell. So in the cytoplasm of the cell, we do have an enzyme that is amazing. This enzyme is a complex structure. In other words, it's simply made up of many different enzymes. So what do we call this enzyme? It is called fat acid synthase. So fat acid synthase is what you can see here. So this fat acid synthase, you need to understand that it has got two parts. It has got the cysteine end and the ACP end. And both of these ends, they are attached to what we call thiol group or SH. It's simply sulfur attached to hydrogen. That's thiol group. So you can see that this enzyme, it is having cysteine end, SH, and also ACP end with SH. So in the first step of fat acid synthesis, what's going to happen is that acetyl-CoA is going to come. When acetyl-CoA comes, it will lose the coenzyme A, and then the acetyl group will remain and will attach to the ACP end. This process is catalyzed by what we call acetyl transacetylase enzyme. In step 2, what's going to happen is that the acetyl group that is on the ACP end is going to move and go to the cysteine end. This process is catalyzed by acetyl transacetylase. Don't confuse acetyl transacetylase with acetyl transacetylase. In step 3, what's going to happen is that malonyl-CoA, which is being synthesized from acetyl-CoA, is going to come in. Malonyl-CoA is a precursor for fat acid synthesis. It's a building block for fat acid synthesis. And you need to know that this is a molecule that inhibits beta oxidation. It inhibits what we call carnitine palmitoyl transferase 1 or CPT1 enzyme, which is for beta oxidation. So this is a very important molecule, malonyl-CoA. It is synthesized from acetyl-CoA by an enzyme called acetyl-CoA carboxylase. So malonyl-CoA is going to come in step 3 and it's going to bind to the ACP end. When it binds to the ACP end, at this point in time, we are having acetyl group on the cysteine end and malonyl group on the ACP end. Now, in step 3, the enzyme that is here is simply called malonyl transacetylase. Now, in step 4, what's going to happen? In step 4, if you look at the enzyme, you're going to find that on the cysteine end, we don't have anything. So at the end of step 4, on the cysteine end, we don't have anything. So what has happened? You're going to find that in step 4, the acetyl group on the cysteine end is going to move and join the malonyl group on the ACP end. Then the malonyl group, it's going to lose a carbon in form of carbon dioxide. The enzyme that is here is called beta ketoacyl ACP synthase. That's the enzyme that is in step 4. Then in step 5, what is going to happen? In step 5, we are going to use this reducing agent that we call NADPH. NADPH comes from the pentose phosphate pathway. So NADPH is going to come there, and then it's going to donate its hydride ion or hydrogen ion. So if you look at that structure that we have at the end of step 5, you're going to see that we have got the OH group. So the enzyme that is here, it is simply called beta ketoacyl ACP reductase. So in step 6, what's happening that we are losing H2O. So the removal of water is simply called dehydration. So the enzyme there is called dehydratase. So in full, we call it beta hydroxyl dehydratase. Then in step 6, what's going to happen is that we are going to use another NADPH. So we're going to use another NADPH, and what's going to happen there is simply addition of a hydride ion. So the enzyme that is here is called enohal reductase. And then what we are going to form at the end of step 7 is what we call butyryl ACP. So when you form butyryl ACP, this is our first fat acid. It has got 4 carbons. So the 4 carbon fat acid that we are going to form is called butyryl ACP. Now, When we form that 4-carbon fatty acid, you can see that those carbons, that carbon chain, it's on the ACP end. It's going to be transferred to the cysteine end so that we can form another new chain there, so that the cycle can repeat. So you can see that we are going to take the chain on the ACP end to the cysteine end. And then the cycle is going to repeat. New malonyl-CoA is going to come there, and everything will repeat starting from step 3. And everything is going to repeat. So that is fatty acid synthesis in a nutshell.